Hello guys, what's up? It's Isabella coming up to you from YouTube channel, Medilab Health. Today we are going to talk about PCR test. By the way, if you haven't subscribed us yet, don't forget to smash subscribe button and click the bell icon. So, let's go to the video. What does PCR stand for? It's polymerase chain reaction. PCR is basically a photocopier. Why I'm telling that is, PCR can amplify a specific region of DNA, and produce large number of its copies. Kary B Mullis, an American biochemist, was the one who first developed this PCR technique in 1985. Not only that, Nobel Prize was awarded to him on this regard in 1993. DNA amplification is such a key factor in many different processes. Literally we can do this in vivo, means inside animals, known as cloning. Actually PCR is the in vitro technique of DNA amplification. We need these things for a PCR to be carried out. A DNA template. This is a selected region of DNA strand which is interested. A thermal cycler, or a PCR machine. This is the equipment which use in this test. And it provides an environment for temperature-sensitive reactions. Polymerase enzyme. This is a type of DNA polymers which has high thermal stability. It was extracted from the aquatic plant Thermus Aquaticus. Primers. As polymerase is a type of DNA polymerase, it still needs a primer to start the proceedings. Primers are short sequences with about 20 nucleotides, and they help to find the exact location of DNA to be amplified. Deoxynucleoside triphosphatase. These are the building blocks of the new template of DNA. Buffer solution. And divalent cations. PCR use in different fields for different purposes. In medicine it used to identify infections occurring from viruses. Apart from that, PCR is commonly used in genotyping, cloning, forensics and paternity testing. Now I'll explain PCR process with an example just for your understanding. Let's assume this is a blood sample of a person who was infected with a virus. First they add some chemicals to remove substances such as proteins and fats. Then the remaining mixture containing genetic material sent to the thermal cycler. The steps of PCR are taking place inside the thermal cycler or PCR machine. Inside this machine, it has small tubes which provide space for the chemical reaction. Actually these reactions take place inside this can be divided into three sections. They are denaturation, annealing and extension. It increases the temperature up to about 92 to 94 Celsius to denature the double-stranded DNA molecule. This will yield bases of single-stranded DNAs. Then primers bind with the complementary bases through hydrogen bonds. This reaction takes place in a slightly lower temperature, around 55 Celsius. Then polymerase starts to add complementary deoxyribonucleotides to the 3' end of template strand. This happens in about 72 Celsius temperature, which ideal for the polymerase activity. In such a PCR produce millions of copies of desired genes within a small time. There are different types of PCR techniques, but RT-pcr, or reverse transcription PCR, is the most popular one, I should say. This convert RNA into its complementary DNA. You might already know that reverse transcription is done by an enzyme called reverse transcriptase. But here it is accomplished by the same polymerase enzyme by altering the buffer conditions. And finally it measures the number of complementary DNA copies it makes after a certain period of time. This is called qualitative RT-pcr. So I think you got some knowledge about PCR technique. Please read the description if you have any doubts. Comment us. Share this video and subscribe for more. So that's all from here. Let's join from in another video.